Okay, so we're in the home stretch of this lab now. We have heat shocked our cells. They have taken up our plasmid. We have added SOC and allowed them to recover. And so the idea there is that the calcium chloride, the heat shock, these are relatively stressful things to do to a bacteria. And so we're providing them with SOC and we're giving them 45 minutes to recover from that traumatic treatment. And so at this point, we need to plate our bacteria onto LB agar plates. And so if you were here, you would have a stack of 10 plates in front of you, and it would be very important to go through and label on the agar side of the plate. You would be listing the conditions and the bacteria that are being plated. So you would talk about how this was a kanamycin or an ampicillin or both plate, and then you would talk about whether you were adding tube X, Y, or Z. So I'm just going to show you briefly how the plating would work. There are a couple of unique things about this that are worth talking about. So in concept, it's very similar to what we did with sterile technique. We've got a flame. We're trying to make sure that we're moving just the bacteria that we want around. And so what we do in order to make that happen is we have a cell spreader that looks something like this. The cell spreader gets submerged in ethanol. And so the important thing here is that the ethanol is what is sterilizing the cell spreader. The next thing we're going to do is pass the ethanol over the flame, and maybe you can see it burning off. That passage over the flame is really just burning the residual ethanol away. And so what's sterilizing the spreader is submerging it in ethanol, and passing it over flame is just burning the ethanol off. I often see students holding their cell spreader over the flame, trying to make it super hot. That's bad practice. So the idea here is that we're sterilizing using ethanol, and then we're using the flame to burn the ethanol off. So we're going to move 100 microliters of cells. So I've got my tube of competent cells here. They've recovered. I can pipette 100 that was not the first stop. I can pipette 100 microliters out. I can go down to the second stop to expel all of that onto the plate. I'm now done with this tip. I can submerge my spreader into ethanol. Again, that's sterilized it. Now I will burn the ethanol off. I'm gonna wait just a second to let it cool off. Touch it to the corner of the agar where the bacteria are certainly not. And then just kind of push this around to move that media and the bacterial cells evenly across the plate. So even across the plate is an important concept. So now we've got our bacteria plated. We flip that over. We can move these plates. So remember, you would have a stack of 10 plates with various conditions and various types of cells. We would move that whole stack into our 37-degree incubator, and we would leave those plates overnight. So at this point, I want to talk about what you're responsible for your lab report. And so what we'll do as part of this online experience is we'll make sure that you have some cells or some plates to count. And so what you'll do is you'll count the number of colonies on those plates, and you'll assemble the colony counts into a table that looks something like this. And so here I've got the different tubes, so X, Y, and Z. Tube X has our recombinant or our potential recombinant. Tube Y has P amp. Tube Z has P can. And then over here we've got our different plates. So LB plus amp, LB plus can, LB plus ampicillin and kanamycin. And then here is just LB with no antibiotic. So we would expect some things from this, right? We would expect growth here. So tube Y on LB plus ampicillin, we would expect cells to grow. So we could say, yes, we would expect growth. What I've listed here is TMC. And so down below the table, I've defined that acronym. So TMC is too many to count. You can use any acronym you would like to put up here. Just make sure that the acronym is defined below the table here. And so too many to count What we're saying here is that there are over 300 colonies on that plate. So over 300 colonies, we're at a place there where there's definitely growth. We don't need to go through and quantify that growth. Too many to count is sufficient for this. And so we're expecting lots of growth here. We're expecting too many colonies to count. We're expecting no growth here. So if the bacteria have been transformed with PCAN, they don't have the ampicillin resistance gene, which means they would be susceptible to treatment with ampicillin. So we would expect growth here. We would expect a lot of growth. We would expect no growth here. We'd flip-flop that down here with the LB-CAN, right? We would expect the PCAN, bacteria with the PCAN would grow in the presence of kanamycin, but bacteria without the kanamycin resistance gene would not be capable of surviving in the presence of kanamycin. So down here, where we've got both ampicillin and kanamycin, we would expect no growth and no growth. Okay, so these are appropriately thought of as our controls. So what we're doing here is we're checking to make sure that the ampicillin and the kanamycin are behaving in a way that we would expect, right? So these are the results that we want. This tells us that we have the plasmid that we expect. It tells us that we have plates that are working effectively. So these are controls, but they're an important part of your discussion, right? In this column, we've got tube X. So these are our potential recombinants. So not all of these bacteria are going to have the recombinant, but some of them, we hope, will have a plasmid that is capable of conferring resistance to both kanamycin and ampicillin. So here, in the presence of ampicillin, we would expect a certain number of colonies. And these should be countable. So you should be able to go through and count the number of colonies that are present here. Again, if we go over 300, you can move to this TMC nomenclature, but I would expect that you should be able to count the number of colonies here. Likewise for LB-CAN. So we should be able to count the number of colonies here. So in each of these cases, we have our potential recombinant, but we also have plasmids that are capable of reforming the original PAMP or PCAN. And so maybe some of these that are resistant to ampicillin haven't made the recombinant, but they're capable of surviving because they have that ampicillin resistance gene anyway. When we get down here, you have to have both, right? You have to have a plasmid that is resistant to both ampicillin and kanamycin. And so that's going to be kind of a rare event. So we would expect to see fewer colonies here than we see with the other two plates. And so 30, maybe that's a reasonable expectation for this. So what you're going to need to do, we're going to provide you with plates. We're going to provide you with pictures of plates. You're going to need to go through and count the colonies on each of those plates. And I'll just remind you that each colony is an instance of one single bacterium that's replicating or dividing to create a colony or a collective on top of that one single cell. And so the idea here is we're counting the colonies and we're assembling this table. The last thing that I haven't talked about is this item at the bottom. So without any antibiotic, we would expect to see a lawn. And so here, we're gonna see cells that have kind of all run together. There are not gonna be clear colonies. They're not gonna be clonal individuals. So it's gonna be one consistent sort of smear or spread of bacteria. The reason we do this LB plate, so the reason that we're doing this, even though we're expecting a lawn, is because what we're doing with the calcium chloride and the heat shock, even though we're allowing those bacteria to recover, what we're doing is a fairly stressful treatment. And so it's possible that we could be killing the bacteria as part of this procedure. And so we do this LB without antibiotic in order just to check to make sure that our bacteria are still healthy and capable of reproducing. So that's the whole idea. So you will need to make counts of the colonies where you can. You'll need to fill out a table that looks something like this. This will be table 1. This will belong in the lab report that's due on Sunday night. And so you will assemble table 1. The other piece of data that you'll need for your lab report is figure 1. And so figure 1 is the gel image that y'all generated before we went on spring break. And so this is the gel image that we made as part of lab number 7. And so what happened here is we have our size marker in this lane. In lane A, we have uncut P amp. In lane D, we have uncut PCAN. And then lane B and C, these are each digested with BAMH1 and HND3. And so what you should see on your gel in lane B, you should see a large band and a small band. We know the size of these bands, so we can refer to them by number. So this large band should be the 3755 base pair band. This small band should be the 784 base pair band. You may see that there are some kind of hazy lines up here. We can say that these hazy lines are most likely uncut plasmid. And the reason we can draw that conclusion is because the bands are at a similar location in lane A and lane B. So by comparing lane A and lane B and seeing that they're in a similar location, we can say that this result, this product of the digestion, is most likely undigested plasmid. And so that kind of makes sense, right? Because we only let our digestions run for 30 minutes. We took a small aliquot out, we loaded that small aliquot on the gel, and then we returned our digestion back to the 37-degree incubator. And so they continued to digest after we started running the gel. And so we would expect to see some undigested plasmid up here. So we can extend that conversation to lane C. Likewise, at the top, you see there are some bands up here that match up with what we see in the uncut lane. We can, because they're similar in both lanes, we can assume that this is uncut plasmid. These two bands down here, 2332 and 1875, these are our specific products that we're hoping would come together to make that recombinant. And so to add a little bit more detail to that, what we're wanting for our recombinant is we want this 3755 base pair fragment to come together with the 1875 base pair fragment. I'll remind you that both of them have sticky ends, so there's BAMH1 and HND3 on either side. They should be capable of sticking together to one another. And the nice thing about this is combining 3755 and 1875 together would produce a plasmid that is both resistant to ampicillin and to kanamycin. Okay, so just as you've been doing for the rest of the semester, you're going to assemble a results section where you have table 1 and figure 1. There need to be paragraphs that precede each of these pieces of data, right? So a paragraph that precedes the gel image, a paragraph that precedes the table. They need to be properly labeled. So table 1, figure 1, they both need titles. The tables are going to have the titles on the top. The figures are going to have the titles on the bottom. This figure needs a figure legend to be associated with it. So make sure that you stick to the rules that we've been using all semester. This is not a deviation. You'll also, as part of the lab report, need to make some predictions in the introduction. And so you're going to make predictions about where you would expect growth. So sort of what we talked about over here, where you would expect growth after you've completed the transformation. And then as part of the discussion, you'll come back and address those predictions. Were you correct? Were you not correct, and why? So I'll remind you there are a number of questions at the end of the lab report. So if you observed colony growth on LB amp plus CAN, this suggests that the bacterial cells are resistant to both ampicillin and kanamycin. How could you confirm that the resistance is due to the presence of the desired P-amp CAN recombinant molecule? So that's a question. You need to answer that question as part of the lab report. There's another question that talks about, is there another way that the cell could be transformed and be resistant to both ampicillin and kanamycin? And so you'll need to think creatively about some possibilities that could help answer that question. So as per usual, I would recommend looking back at the rubric again. So we are grading your lab reports based on the information that's laid out in the rubric. Make sure you're checking all of the boxes on the rubric, and then make sure you're answering the questions at the end of the lab report. And I'll remind you that we're available to help. I know that these are extreme circumstances, but if you have a question, please ask it. I'll also warn you that if you send an email to an instructor individually, we've all been instructed to put those emails or put the questions from those emails into the discussion board that's associated with each lab. So your words may end up being communicated to the whole class. The idea there is we're trying to make sure that we're not being redundant with our time. So I hope all of this makes sense. I hope that the lab reports that come to me are high quality. I want you to know that we want you to do well. We're here to support you. Please reach out if you have questions. Thanks, y'all.